In this video, we will continue to discuss some of the results of isoferulic acid on pancreatic cancer cell lines, that is PANC1 and BXPC3. In the previous video, before we discuss our results, in the previous video, I have sometimes mispronounced this proponium diiodide and annexin 5 assay as Comet assay. This is not the Comet assay, this is the assay to check the dead cells using proponium iodide and annexin 5. So when the cells are dead, the annexin 5 can penetrate inside the cells. That's why we see annexin 5 positive cells. So now let's just move into this tunnel assay and comet assay. These assays are also made to check the apoptosis in the cells. So first we discuss the results for the tunnel assay, which is on the left hand side of the figure. So what they did is they stain the cells with this tunnel reagent and keep it, and they stain the cells with tunnel reagent for 48 hours and then check the images, and the pink fluorescence image, or this fluorescence image will shows that these cells are in these cells the apoptosis has been triggered, or these cells are the apoptotic cells. So we can see that in the control the amount of apoptotic cells are less compared to the case where isoferulic acids are used in low concentrations, and this tunnel staining signals further increases when the isoferulic concentration increases, and it goes to a maximum level where isoferric concentrations are higher, and here the high means that 12.5 micromolar isoferulic acid is used. And same things they have observed for the BXPC3 cell lines. For the controls they see the low signal of the tunnel staining, and for this low concentration of isoferulic acid they see a low concentrations or slightly higher concentration compared to control, slightly higher signals compared to control of the tunnel staining. And for this isoferulic acid at medium concentrations, or that is 6.25 micromolar concentrations, they see some increase in the tunnel staining, and for this high concentration of isoferulic acid, that is 12.5 micromolar, they see a further increase in the tunnel staining signals. So this indicates that or further substantiate the previous finding that this isoferulic acid will trigger apoptosis in a apoptosis in the pancreatic cancer cell lines in a dose dependent manner. Next, they also did this comet assay, and before going to the comet assay results, that is in the panel B, let us understand their hoist staining. Hoist staining is used in this experiment to check the effect on the nucleus, and what they are able to see is that when they use isoferulic acid, they see some condensations of the chromatin and DNA damage, which is marked by this white arrow, and this condensations of chromatin and DNA damage increases with increase in the isoferulic acid concentrations. We can see that the white arrows are low in the low concentrations, and it increases with increase in the isoferulic concentration to 6.25 micromolar, and it further increases when the isoferulic concentrations is high, that is 12.5 micromolar. And same things they have observed for this BXPC3 cell lines, that in this case in the control also they see some condensation of chromatin and DNA damage, but compared to the isoferulic acid treatment, these condensations and DNA damage signals in control are lower. Here we can see that with the isoferulic acid treatment there is an increase in the condensation and chromatin, condensation of chromatin and DNA damage. And when I did the comet assay, here we can see that in the case of control, the smearing of this DNA is low for the PANC1 cells and also for this BXPC3 cells, but with increase in this isoferulic acid treatment, or with the isoferulic treatment, the smearing of this signals is high, indicates that these DNAs have this tail end, or these DNAs are damaged. And same things they have observed for BXPC3 cell lines. And with increase in the isoferulic acid concentrations, they can see that the tail DNAs or smearing of this DNA increases with highest for this high isoferulic acid concentration tested, it is 12.5 micromolar, and this is true for both BXPC3 cell lines and PANC1 cell lines. The quantifications are shown on the right hand side, where we can see that with increase in the dose dependent increase in the isoferulic concentration, there is an increase in the tail DNA percentage for both PANC1 cell lines and also for the BXPC3 cell lines. So this results along with this uh, annexin 5 and proponium iodide results indicates that this isoferulic acid can trigger apoptosis in the pancreatic cancer cell lines. That has been tested in PANC1 and BXPC3. So I hope you understood or might be convinced to some extent that isoferulic acid cause apoptosis or apoptotic or apoptosis. Isoferulic acid cause apoptosis in the PANC1 and BXPC3 or pancreatic cancer cell lines. That's all for this. That's all for this video. If you want to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.